Hey everyone, I wanted to take a few minutes and just talk pretty briefly on protein synthesis and mitosis. I just want to make sure that you feel comfortable enough going into our upcoming unit test on these topics. So, first and foremost, we're going to talk about protein synthesis because protein synthesis kind of brings together all the information about organelles because it takes a lot of organelles working together to make a protein. So I want you to keep that in mind for the big picture. So first and foremost, proteins are coded for by genes in your DNA. Remember, DNA is found in the nucleus, but proteins can only be made in the cytoplasm. So we got a little bit of a situation there. We have to get the information that's in the nucleus on the DNA, and we have to get it out into the cytoplasm where we can actually make a protein. But the cell has a plan for this. So we're going to see that the nucleus, the ribosomes, which by the way, just a reminder, they're made in the nucleolus of the nucleus. The endoplasmic reticulum, the Golgi, and vesicles are all going to play an important role in protein synthesis. So that's a lot of organelles. And if I'm really honest, cytoskeleton is going to play a role there. Like lysosomes play a role because they break down proteins when they get old. So really all of them do in some way. But those are like the big ones when it comes to making proteins. Now, big picture for protein synthesis, there are just two major steps: transcription and translation. Transcription happens in the nucleus. Translation happens in the cytoplasm. So transcription is when we make an mRNA copy, M stands for messenger, of DNA in the nucleus. So whatever gene is coding for the protein the cell wants, the cell is going to make an mRNA copy of that gene inside the nucleus, and then the mRNA is going to exit the nucleus and go out to the cytoplasm because it has the directions for how to make the protein. So why do we have to do this? Because DNA can never leave the nucleus because the nucleus is the only safe place. There aren't DNases in the nucleus that will chew up and destroy the DNA, but the cytoplasm, dangerous neighborhood for DNA. So when cells need a particular protein, maybe an enzyme, it will make an mRNA copy of that gene from the DNA that's inside the nucleus. So now we've got that mRNA, like I said, that has a set of directions for making that protein, which really just means what amino acids and in what order. And that mRNA is able to exit through a nuclear pore to leave the nucleus and end up out in the cytoplasm. mRNA is only able to exit the nucleus through the nuclear pores because they have the secret password that lets them get out. Most things cannot move into the nucleus or move out of the nucleus because they don't have the molecular password to do it. Okay, so what does transcription look like? Well, again, can't emphasize enough. It happens inside the nucleus, and the goal or end result is the production of a messenger RNA. In order to do that, we're going to have to uncoil the DNA and unzip it. And just a reminder that DNA is held together with hydrogen bonds that we learned about earlier. And once we've unzipped the DNA, we can make a complementary copy of one strand of the DNA. So that whole A and T, C and G thing that we talked about, that's how we make a complementary copy. But remember, in RNA, we have uracil instead of thymine. So any place you would think there'd be a T, it's going to be a U instead. When we first make an mRNA in the nucleus, it's called a pre-mRNA because inside the nucleus, that mRNA is going to have two pieces added to it. They're called a 5' cap and a poly A tail. And basically, those stabilize the mRNA so it doesn't fall apart and also allow it to exit the nucleus through the nuclear pore. Once that mRNA gets out into the cytoplasm, it's going to attach to a ribosome. That ribosome could be in the cytoplasm, or it could be attached to a rough endoplasmic reticulum. In either way, at the ribosome, the mRNA is going to sort of be fed through the ribosome, and the ribosome will assemble the amino acids based on the messages in the mRNA, and those amino acids will be put in the correct order, and they will be covalently or peptide bonded together to create the polypeptide that will become the protein. The way the amino acids get to the ribosome and to the mRNA is because another kind of RNA called tRNA delivers them. So each tRNA is unique in that it will only hold one specific amino acid, and the amino acid it holds is the one that the anticodon codes for. So the mRNA strand, every 3 nucleotides is a code for an amino acid, and the anticodon on tRNA matches that codon. And so each tRNA carries just one type of amino acid that it will bring and deliver to the ribosome to place in the correct location on the mRNA strand. So that's how we get the amino acids, the right amino acids in the right place. And then, assuming that ribosome is attached to rough endoplasmic reticulum, as we build this protein or the polypeptide, it will go into the rough endoplasmic reticulum, and then it will get folded inside the rough endoplasmic reticulum because that's a very safe place. The temperature's right, the pH is right, the salinity is right, so we shouldn't have any denaturation happening there. And then from the rough ER, a vesicle will take that protein to the Golgi body. And then in the Golgi, it might get modified, like a sugar could be added to it or a lipid could be added to it. It will be stored or it will be packed up with other polypeptides or proteins, and then it will be shipped via a vesicle to its final destination, which might be the cell membrane or outside the cell. And that's it. That's protein synthesis. For us, we really just want to know that protein synthesis is how we make proteins. We want to know it has 2 steps, transcription and translation. We want to know that transcription is the making of mRNA in the nucleus, and translation is the making of a protein in the cytoplasm at the ribosome. And we want to have a sense of some of the other organelles that play important roles in that process. If you want to learn more about this, there's a great animation in the study area of Pearson MyLab. So if you poke around in Pearson MyLab, you should be able to find those animations. Okay, and then the other thing that I want to just talk real quickly about, especially if you're not in my lab, is I want to talk a little bit about the cell cycle. So just remember, the cell cycle consists of both interphase and the mitotic phase, or M phase. An interphase can be divided into 3 parts, or 3 stages, or 3 phases, whatever word you want to use. The 3 parts of interphase are G1, S phase, and G2. In G1, the cell is going to grow and do its normal job. In S phase, it's going to replicate its DNA because it's preparing to go into M phase. In other words, it already knows it's going to divide, so it needs to replicate all of the DNA. For you, that means all 46 chromosomes. Then in G2, which is a pretty short cycle, the cell is going to finish growing, it's going to finish making any proteins it might need, and it's going to make any additional organelles that it might need in preparation for mitosis. And as long as everything checks out, because there are checkpoints in this cell cycle, and if the checkpoints get out of control, like they're not working properly, that's what leads to cancer. But assuming that everything is good, that the DNA was copied correctly and there's no problems, then the cell will enter M phase. And M phase is mitosis, could be meiosis, but we're not talking about those kinds of cells in this class. So for us, it's mitosis. And then mitosis would have prophase, metaphase, anaphase, and telophase. And then most of the time, cells after mitosis undergo cytokinesis. So mitosis is the division of the nuclear contents, whereas cytokinesis is the division of the cell itself, or the cytoplasm, if you want to think of it that way. Now you'll also notice on this slide something called G0. This is a phase that only some cells enter because they never divide, so they don't need to be going through this cycle. They just kind of go into this extra special phase called G0. So things like skeletal muscle, neurons, your fat cells, most of these cells never divide, and so they don't ever need to enter S phase, so they just hang out in G0 instead. Now, S phase is special because that's where we're going to replicate the DNA. This is a little different than protein synthesis. Protein synthesis is when we're just making a copy of one gene in the nucleus so that we can make the protein that that gene codes for. DNA replication is when we're copying all of the chromosomes so that after mitosis, we have two genetically identical cells. So they both involve copying DNA, but they have different purposes, and as a result, they look a little different. So again, in S phase, we're going to copy every single chromosome so that we can create two genetically identical cells after mitosis and cytokinesis. Also, we're going to have centrioles that have to be replicated. So in addition to replicating the DNA, we are also going to replicate the centrioles because we're going to need centrioles in each pole of the cell during mitosis. I just want to point out that if you remember from high school anything about meiosis, that meiosis is different than mitosis. Mitosis creates two genetically identical cells, but meiosis is how we make egg and sperm. So they are genetically variable and they only have half the number of chromosomes and you end up with 4 of them. So it's just good to know the differences between them, but we're focusing on mitosis in this class. And so for us, we're going to have the 4 phases of mitosis: prophase, metaphase, anaphase, telophase. And in lab, you should have learned about each of these phases and what things or events happen during each of these phases and what these cells look like under the microscope during these phases. So this slide gives you that information. And then on this slide, you can see some really nice pictures of cells as they go through the phases of mitosis. So notice in this cell, we can clearly see a membrane around the nucleus. So this is not mitosis. This would be interphase, right? Could be G1, could be S phase, could be G2. We can't tell, but we can see that there's a nuclear membrane. So we have to assume that it's interphase. Now, once we move into M phase, we're going to see that the nuclear membrane breaks down during prophase, and we're going to see the centrosomes with the centrioles form the spindle formation during prophase. And by the time we get to metaphase, we can see the separated centrioles inside the centrosomes. We can see the spindle fibers, and we can see the chromosomes lined up down the middle. As the chromosomes separate from the centromeres, that's just what holds the copies together. As they separate, that's going to be anaphase, and the chromosomes will move apart. So we're separating the identical chromosomes, and they're going to opposite poles of the cell. We might also start to see a cleavage furrow here. And then here in telophase, we can see that we're starting to pinch. We're seeing cytokinesis. We're starting to pinch the two cells apart from one another. The chromosomes are in 2 different cells, and the next steps will be that the nuclear membrane will reform, the chromosomes will uncoil and disappear, and we'll be looking at cells that look something like this. Is very late telophase here. We still need the nuclear membrane to come back, and we still need the chromosomes to uncoil for this to be complete. So hopefully this helps you just feel a little more comfortable with the content from lecture in Unit 1.